Hello, my dear students. This is your host for today's class, Dr. Syed Bakar. In this class, let us study about root nodulation of Rhizobium bacterium, which is used as biofertilizers. So this rhizobium is abundantly found in soil. You can isolate this from the soil. But when this rhizobium comes in contact with leguminous plants, example peas, chickpeas, clover, etc., when they come in contact with these plants, they form a root nodulation. This is a specialized condition in order to fix nitrogen, in order to fix the nitrogen in the soil. This fix the nitrogen so that it can be available to the plants and it increases the plant growth. Ultimately, it enhances the plant growth. So let us see how this root nodulation and nitrogen fixation happens. Just imagine these are the roots, and here you have stem, and these roots are said to have root hairs. Root hairs are very minute structure in comparison with roots. Let us talk about this root hairs. If you enlarge this, the root hairs will be like this, attached to the big roots. And what happens here is the bacteria Rhizobium comes and closer to the root hair. The reason is this root hair is capable of secreting phytohormones. These phytohormones might be flavonoids, amino acids, etc. When they come in close contact with this gets ruptured and makes a way opening to enter the bacteria inside the root. So this structure formed to enter the roots is called as infection tube. So the bacteria enters the infection tubes, migrates and reaches the cortex of roots. Here you can see the bacteria reaches here. Once the bacteria reaches the cortex of the root, it undergoes metabolic, physiological and morphological changes, and this multiplies and form a nodule-like structure outside the roots. So let us see what happens now. Once it reaches, it forms nodules, and here this nodule, what you are seeing, contains Rhizobium bacteria which undergo metabolic transformation along with metabolic and physiological transformation to form a condition called as bacteroids. Students, remember this, bacteroids. This bacteroids is a condition where it doesn't multiply, but it will be in an active form. This bacteroids is highly sensitive to oxygen. Hence, they secrete, plant and bacteroids secrete leg hemoglobin. Leg hemoglobin. This leg hemoglobin acts as oxygen scavenger. It removes oxygen, maintains the anaerobic condition. And this bacteroids is also capable of secreting nitrogenase enzyme. This nitrogenase enzyme converts nitrogen into ammonia. So from here, from soil, it goes and attracts to the leguminous plants, and it attaches to the root hair. From the root hair, it causes infection tube. This infection tube gives a path for rhizobium to enter. Once it reaches the cortex, it forms bacteroids and forms root nodules. This bacteroids secretes leg hemoglobin, which acts as a oxygen scavenger to maintain anaerobic condition. They also secrete nitrogenase enzyme, which converts nitrogen into ammonia, which is utilized by plants, which increases the growth. So this is how it forms root nodules. Now let us talk about how are this rhizobium bacteria isolated. As we came to know that they form root nodules and perform nitrogen fixation. This is leguminous plant. You cut off this plant part, and now we are interested to isolate rhizobium, so we are only concentrating on roots or root nodules. So these root nodules are collected and immersed in surface sterilizing agent, then washed with distilled water Then again it is dipped into 70% of ethanol. 70% of ethanol, again wash with distilled water. Then these nodules which have undergone surface sterilization are crushed. Or squeezed in under aseptic condition. Then inside the test tube, under the aseptic condition, these root nodules are crushed, and these root nodules are performed serial dilution. They undergo serial dilution, 10 power of minus 1, minus 2, minus 3, like this, up to 10 power of minus 8. Then it is plated onto the media. And this dilution factor are spread onto the media, on a specialized media, yeast mannitol agar, which favors the growth of rhizobium bacteria. Once you see the colonies growing onto the media, these colonies are subjected to biochemical and molecular characterization along with gram staining. The rhizobium bacteria is gram-negative bacteria. So this was the formation of root nodule of rhizobium with leguminous plants. I have also discussed briefly about how you can isolate a rhizobium bacteria. So let's conclude this video today. See you in my next video. Till then, I wish you all the very best. If you have any doubt, you can call me or message me. Goodbye.